Okay, it's about time to start. Welcome to this symposium. This is a symposium 34. It's organized by our museum. First of all, I want to introduce myself. I'm the first moderator, Ming Lun, and I am from the National Museum of Natural Science in Taiwan. And this symposium is organized by our museum. And the aim is to enhance understanding of the value of museum collections for the research of biodiversity and the significance of citizen science to the museum, conservation, and many other biodiversity issues. And most of all, the most important one is we are seeking for the potential international collaborations. So if you are interested, please contact us, okay. And in total, we will have 10 talks. So they are divided into 2 sessions from now on to the end of this meeting today. Okay, how? And sorry. And before we proceed, we want to thank the primary sponsor of these meetings, like Picture A and Max Communication, Pensoft, and several others. And our first talk is by my colleague. He is a botanist, Alex Yang. He will talk about the museum collection in the biodiversity value in Taiwan. Okay.
Good afternoon, everyone. I'm Alex from Taiwan, as Dr. Zheng introduced. Today I want to talking about the vascular plant in our museum, our herbarium. And this is our museum. Looks beautiful and nice. But the herbarium is more inside. So my museum, the abbreviation is NMNS, National Museum Nature Science. So normally NMNS sometimes difficult to pronounce. So normally I would say no mother, no sister, something like that. So that is NMNS, okay. And the herbarium is TNM abbreviation. Now in Taichung City, Taiwan, so normally for our botanists, we will write down as a TMM. And we established in 1983. And general collection, this is talking about vascular plant. General collection is 275,000 vascular plant. And we also including non-vascular plant like Moses and the RG and something else and others. Then about 36,000 only. And fungi, someone we were talking about only 38,000, something like that. Totally about 351,000, that is total. But we are very young, as you know. So not like in Taiwan, like a TAI, National Taiwan University, and the TIF. They got about 80 years or 100 years. So, but however, we are the richest the third in Taiwan in our museum. So, and what we have special have, we have some type specimens, so not many very, very old or famous one. But however, because my major is climatics, so we do have one something like a collect in China, 1886 or something else. It's the oldest one in that they exchange to us. So it is the oldest one. And we do have some Solomon Island collection. And there is some real area and not many specimens in other countries. So we do have such project in 5 years. And we do have some people study Musa. So we do have some, like this is type specimen. And this we do have one chance. We do. I have a meeting with some people from North Korea. After that, I know, oh, it is real. The very real specimen from North Korea. So some South Korea people visit us to digitize. So for those specimens, we do have about 200 and more. And yes, this is Solomon Islands specimen. we do have about 15,000 or something more. And for our collection, we will put the map on the label, so easily to know where it is. And we do have a Yano specimen, and this is just my topic talking about Yano specimen, because Yano is a Japanese, and he's a plant hunter, and he collect for Makino, Professor Makino. So at that time, we do have a such project talking about type specimen figure reconstruction of Taiwanese vascular plant, that the international cooperation project of digital contents integrated and cooperated. So at that time, I choose some European herbarium, and this I visit LE St. Petersburg in 2010. So at that time, I find some type specimen. Also, I try to find some specimen. Then I found Yano specimen. So then, oh, okay. So this is Makino specimen. Makino, he has visited Taiwan only about 3 months during November and 1896. And for those, we just got this about 100 specimen from TI University, a duplicate sent to us, and also many bamboo. He's a bamboo, very interesting. And now we back to Yano's story, because the Yano is what I talk, and quite special. And we also connect with Japan, some herbarium. And the one is I found the first one. So I found this specimen collected by Yano in 1897 in Green Island. So I took a picture image when I go back to ask some Taiwanese, my teacher, my supervisor, and some Japanese teacher talking about who is this person, because there's no any record talking about Yano, this person collect in 1896 to 1897. At that time, just Taiwan changed. Japanese come to Taiwan, and Taiwan is Qing dynasty just changed. So at that time, lots of foreign people went out, and Japanese not coming yet. So why these people will come here? So it is quite mystery. So then, when I go back, we try to find a project. 2009 July to 2012 June, and at that time for this project, for me it's quite exciting, because this project I have to be there only during, because I have a lecture in the university, so I have to be there during winter time. For people from Taiwan, I don't know, I cannot image about -20 at that time to St. Petersburg. I don't know what kind I should wear. So it is, when I saw this, oh, snow, that is snow, but it's really cold. This is what I first to visit St. Petersburg in June. So, and the next project is going to winter in January. So, it's quite totally different. I have joined there at Huainan and also Huaidade, something like that. So, and this is why we work at there. And St. Petersburg, there's Komarov Botanical Institution. But so Komarov, in the Komarov, we know is a famous person, and also why Yano, this person, he why they collect such specimen from Taiwan. So we after this project, we're talking about with Demetri and Alisa and Irina. This is what we have worked together. Because in the botanical garden, there's only one status. I think that should be Lenin, not. I think whether he's Komarov, because it should be. But however, there's a Komarov statue only in the building, not outside. So look totally different. So for this project, after that, we do have some exhibition talking about this story, mystery person, because until now, no one knows what Yano looks like when he was born, when he was passed away. No one knows, no any record. So that is quite interesting. So at that time, so this person, Dr. Gilmerty, at that time he's a deputy director, but now I don't know whether he's hired or not. When we invited him to be a guest to introduce this project, he said when he was young, he know Taiwan, but they belong to another world. For Russia, they cannot. At that time, it's a USSR, not Russia. So he think Taiwan mean you could realize. So, but many years ago, he came to Taiwan, stand in Taiwan. So for him, it's so exciting for all of them. So this is why we have a exhibition. At that time, they're talking about him and also some history. Okay, and Yano collection, we do have a, lucky in St. Petersburg, I got 293 taxa, including fern, conifer, and flower plants, and belong to 393 different species. And we also got 200 duplicate to our herbarium. And also, we do have such exhibition. We found one person, Matsumura, he published 1902. He said some plant, this one, talking about some Yano collection in Hokkaido University. Why I'm saying I'm lucky? Because during the LE specimen, I found that is the number is Yano collection. So 1, 2, blah, 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 about 500. So I got a list because this one is not many. Matsuma is only 113 taxa, 140 specimen. So I got a list, so I can check according to the list. So I go to find some other different species. So I find more after that. So I found, this is published in his paper, but I found 127 taxa of 228 specimen. Found more specimen there. So make it Yano collection is more clear. This is, I worked there, Hokkaido, and this is Yano, the size is much bigger, so we separate the specimen. And the orchids, then finally we found Yano, this person, he's the first person collected in Lanyu. There's a different small island, and at that time, so there's a moss. And also lots of Green Island and some small, but he also collect in North. And also this, my colleague has collect pick some material. He can also analysis for the DNA barcoding. He told me can analysis. Some older real species now, endangered species, at that time very common because I say he just a plant hunter. Okay, and finally we found one specimen, and this one, Leishmania candida. So this one is when I was in St. Petersburg, I can't identify because according from Taiwan, the first and second editions, I can't identify. So this one should be come to Taiwan earlier, so duplicate. Then 2 years later, we found this one we identify Leishmania candida. This is a new record to Taiwan and also extinct. So extinct specimen or species in Taiwan, so but however, herbarium can keep such a secret, such very important thing in herbarium, waiting for someone to study. So you can find some 100, 200, long ago, the specimen, what it looked like. Okay. So now we do have, so according, if you want to know Taiwan during more than 100 years ago, the vegetation, we can, according to Yano collection and also Makino collection, perhaps you can get some picture, know what it looked like and what different from now and that time. So I think it's a good, can do more work in the future. Thank you very much.
Thanks, Alex. We have a couple of minutes. Any comments or questions for Alex?
Thank you very much.
We are all nervous because of language. Anybody? Or okay, thank you.
Okay, thank you very much.
So we change to our second moderator, Yulin Huang.
So good afternoon, everyone. So I'm Yulin Huang, also from the National Museum of Natural Science. The next speaker is also our colleague. He's the mycology collection manager, Dr. Tdwg Chen. So let's hear his talk.
Hello everyone, thank you come to my talk. And I'm Tdwg Chen from National Museum of Natural Science in Taiwan. Today I'm going to share the topic exploring fungal diversity in East Asia, insight from fungal collection in our museum. As you know, fungi is very diverse. You can see the image. You could see different color, shape of fungi. So now, with an estimated 2 million species are recorded worldwide, they play important role in ecosystem and human life. However, many fungi remain unknown to science. So where are these undescribed fungi? They could be preserved in collection but not yet studied, hidden in understudied regions, or masked as cryptic species, or existing in an environment without obvious structures. Discovering these fungi is important for learning more about biodiversity and maintaining ecosystem balance. With this background in mind, let me briefly introduce our institute, which plays a key role in studying and preserving fungal diversity. The National Museum of Natural Science, located in central Taiwan, was established in 1986 and has become a popular place for all generations. Our museum hosts significant collection in geology, rheology, anthropology, botany, and mycology. The herbarium code for our botany and mycology collection is TNM. We have over 38,000 fungal specimens, with about 10% of them having living cultures. TNN's fungal collection includes specimens from 49 countries, with focus on East Asia, with about 26,000 specimens from Taiwan and about 8,800 from mainland China. We have more than 320 holotype specimens. About within those holotypes, 173 holotype was named by TNN's mycologists, including myself. So you can see it's our Fungarian's photos on the right. Let me introduce 3 famous species named by our mycologists, all of which are wood decay fungi. On the left is Gynoderma linzi, also known as reishi in Japan. It's widely used in traditional medicine in East Asia. The holotype is from Hubei in China. On the right is Sanhuangnoporus sanhuang, commonly called sanhuang in Chinese. It's another medical fungus with its holotype from Jilin province in China. Finally, you could see Mycena cyanocephala. It's a very cute and tiny fungus from our botanical garden in museum. You could see the image. It's also the symbol for the fungal kingdom on iNaturalist. Nearly all of our fungal specimens, 99%, are now digitized and accessible online. You can explore our collection by scanning the QR code to access our database. Just enter the detail and filter the results. The image of the specimen will appear first, making it easy to browse. You can also switch to a list view to see all specimens in order. From there, you can select specific specimen to view more detailed information. Along with basic information, we also show whether each specimen has a culture or DNA available. About 11% of our fungal specimens have DNA sequence, providing valuable data for research. Now let's talk about collection-based research. On the left-hand side, you could see the image. We collect fungal specimens from various ecosystems and substrate across East Asia. After collection, we conduct morphological and molecular identification and isolate living cultures if possible. These specimens and strains are then stored in our museum, serving as a valuable resource for future studies on fungal diversity. Now I would like to share two of my research projects that use TNN's fungal collections. The first study focused on the systematic of the genus Paratrichepton, which was published in Fungal Systematic and Evolution. The genus Paratrichepton includes a single species, Paratrichepton acuratum. It's a rare wood decay fungus found in East and Southeast Asia. It grows on the trunks of broadleaf trees in old forests. For over a century, there were fewer than 10 records for this species from these regions. You could see the blue dots on the image. Each image represents one specimen. The type specimen of this species was collected in Sumatra, Indonesia, with additional records from Taiwan and Japan. In 2017, during a field trip to the Vietnam border, I rediscovered this species and obtained its DNA sequence for the first time. On the right, you can see a phylogenetic tree reconstructed from 5 genes. My research confirmed that Paratrichepton belongs to the older Gloriophyllades in the Agaricomycetes, and it's a new record for China. The second study explored the species diversity and multi-gene phylogeny of Brabioid clay within Polyporades. This work was published in Fungal Diversity. You could see the image. The Brabioid clay in Polyporades consists of wood decay fungi with diverse fruiting body forms, including resupinate fruiting body, pilate fruiting body. On the right, you can see a phylogenetic tree based on 5 genes. The clay is divided into 3 families, Phanerocataceae, Apicaceae, and Merliaceae, shown in 3 colors. We identify totally 57 genera, including 6 new genera and 26 new species. Most of these new species were found in tropical and subtropical regions of East Asia, but some were also discovered in temperate area. Let's see example. One of the exciting discoveries is airbowl Ephibula bambusicola. It's a new genus and species found on bamboo in high mountains of Taiwan. On the right, you can see beautiful microscopic illustrations of this species. In conclusion, the National Museum of Natural Science fungal collection are a rich resource for exploring fungal diversity in East Asia. Through digitization, accessibility, and research, we continue to uncover new taxa and deepen our understanding of fungus' role in ecosystem and human life. These efforts not only highlight the importance of collection-based research, but also contribute significantly to the global knowledge of fungal biodiversity. Thank you for your attention, and special thanks to the following agencies for their research funding. Anze Zichen, feel free to ask any questions. Thank you.
Okay. So we have a couple minutes for the questions. So anyone has questions?
So what fungi you can find in mangrove forests?
You could find some wood decay fungus growing on mangrove forests, or you could also find some aquatic fungus living in water.
Any other questions? We are 3 minutes left for the next talk, so maybe we can just exchange yourself and come back at 3 minutes. Oh, it's now 2 minutes now.
Okay, thank you.
Okay, it's 3 o'clock. So the next presenter is myself. I'm Yulin Huang from the National Museum of Natural Science, and I'm also working in the mycology division. So today I'm going to talk about the cryopreservation of fungi in our museum. So my colleagues already introduced our museum, so I just briefly talk about that. So we are museums in Taichung, Taiwan, and which was opened in 1986. And so far we have more than 1 million natural history collections. And for the mycology division, we have more than 38,000 fungal specimens, and our herbarium code is TMM. So here are our fungal specimen preparation and registration workflows in the museum. So the top lines in black text shows the general preparation of the dry specimens for fungal specimens. So we collect the samples from the field trip or the collection project, and then we send the mushrooms in the oven to dry, and then followed by the microscopic identification, and some of them will do the molecular identification, and the specimen itself will be packed by our volunteers or the technicians. Then the data will enter into our database, and the label will be printed out and attached to the small packets of the mushroom packet, and then we send it to the -30 freezers to prevent the insect in the specimen, and then put that into the herbarium. And these are for the dry specimens. And today I'm going to talk about the preservation of the fungal living culture. So we have to isolate the living cultures before they die. So before we put them into the oven, we can either do the spore print collections and then grow the spores on the medians to subculture them, and then we can do the preservation of the culture, or we can directly isolate the mycelium from the fruiting body or from the substrate like the wood, and then we culture this mycelium and we can get the living cultures. And before we send the cultures to the cryopreservation, we also do the molecular identification to double check it's the right fungal strains. So here are some methods for the fungal preservations. So here are the general vessels, and usually in the mycology lab, we do the serial transfer. So we only need the fungal strands for a few months. So we can just put them on the culture medium and grow it for several months and then transfer to another petri dish. So this just for the short-term storage. For long-term storage, because the log cost is very important for the general lab practice. So the distilled water status is the best way to store the fungi in the room. It can last about 2 or 3 years. But from our experience, the water stasis or water voucher can preserve the fungi for 10 years and is still alive. And another low-cost way to store fungi is the Miller oil. So you just put the oil onto the slant culture medium and you can store them. But this method requires a larger space because you have to make sure that all slants are stand up. So it requires a large space. And another common way is the freeze-dry method. The freeze-dry method requires the machines to dry the fungi, and it's not suitable for all the fungi, so it's not the best way to do the preservation. And silica gel is also a very cheap way to store the fungi, but sometimes it cannot guarantee the viability of those fungi. So the better way is to store the fungi in a low temperature, and usually the standard freezer is not ideal, although it's a low-cost way. So the ultra-cold freezer or liquid nitrogen system will be better to store the fungi for the long-term storage, or for, we can say, the permanent storage. And our museum uses the liquid nitrogen vapor system to store the fungi. So here is some of the cryopreservation of fungi in our museum. So our cryopreservation starts from 1996. It's about 10 years after our museums opened. So currently we have more than 5,000 fungal strains in our collection. And for each fungal strain, we have the triplicate of each strain. So first we have the liquid nitrogen storage system. So we put that into liquid nitrogen. And then we have one copy of that strain to back up, store in the -80-degree freezers. And another one is the still water, so at just 15 degrees. This one is just for the easy accessibility for us. If we want to use that fungal strain, we can directly take it from the fridge. We don't need to recover the fungi from the liquid nitrogen. But sometimes it fails, so when it fails, we have to go to the liquid nitrogen system. Okay. So from our test, we found that the fungal strands preserved for more than 25 years and still alive and can be recovered from the cryopreservation vials. And here is how we do the preservation. And we made a video of this process for our exhibition several years ago. If you are interested, you can scan this QR code and you can see the whole progress of this cryopreservation. So briefly, we add the cryoprotectant to the small cryo vials. And the cryoprotectant contains the dextrose, trehalose, dihydrate, and glycerol. And these three protect the cells from the freeze environment. And the PDB, which is a general medium we culture fungi, the potato detritus broth, it provides the nutrients to the fungal cells. So, and then we transfer the mycelium tubes into the cryo vial and immerse them for 1.5 hours. So we cut them into the small cube, put in the tubes, and then transfer the mycelium, put that tubes. And we stand for 1 and 5 years because we want the cells to absorb and to immerse in the cryoprotectant. And then we use the control rank cryo freezer to freeze the cells to -70 degrees, and then move the fungal cultures to the liquid nitrogen vapor storage system or the -80 freezer. And for the control rate cooling system, here is our temperature program. You can see in general, it's about decrease 1 degrees to 1 degrees every minute to slowly the freezing time. But here you can see a steep drop here because when it reaches the -40, it may create the ice crystal in the cell. So the Krebs step can pretend and can protect the cells and make it increase the stability of the cell. So overall, here is the collection summary of our museum. So we currently have 38,000 dry specimens and 5,000 living cultural strands, and about 4,500 fungal strands have the records connect to the dry specimens. And from them, there are about 2,600 or 700 strands also have the DNA data. So they are linked. And we have another DNA database. It contains 4,700 DNA solutions. And from them, there are more than 8,000 loci are sequenced. So for to manage those 3 different types of collections, we have collection management systems, which was migrated to the new system 2 years ago. So previously, we only have the fungi collection management system, and all the fungal cultures and fungal DNAs are kept in the Excel sheet. So it's not a very ideal way to manage the data. So 3 years ago, we just put them all together. So here is our system. So this sheet showing the fungal cultures information. We can see the collection number and the related fungal specimens category number, and also where the isolate from. For this case, it's isolate from the as the fungal endophyte. And also we can see the storage position, so we can get the fungal strands in our storage system. And also we have the culture status to measure its normal or its death. And it also has the DNA sequencing data. So you can see it has the 4 different sequences in mind from the different loci, like the ITS or the TB1 alpha. And recently, we start to take photos of our culture collections. So some of our new collections has the photo of the culture too. And if we want to see some specimens, if they have the fungal culture collection or DNA data, we can simply search them in our system. So here I enter the genus names of the mushroom, and then I can see the list of the mushroom, and we click that, we can see the detailed information of the collection data, and also I can assess if the fungal culture is on this number, and also have this DNA number and which gene was sequenced. And for the DNA data, we can see the sequence of the fungal strands, and also if it's already update to the gene bank, we can see the gene bank accession numbers. So the collection values from our museum, we think that the cryopreservation system can make the living fungal diversity available for future applications. And this comprehensive database can enhance the accessibility and the usability across the museum's fungal collections. Okay, so thank you, and I'm happy to take questions. I think we have a couple minutes for questions. So any questions? Then we can also have like 4 minutes for the next talk. Thank you.
Yeah, thank you. Okay, it's time. Our next and the last speaker of this section is Dr. Zhang Weilin, a herpetologist from National Taiwan University.
Hello, everyone. I'm Jiang Wenlin. I'm from Taiwan, and I'm so glad to see you guys here. It's my second time to join Spnhc meeting, and it is good. You'll know it later because last time I get some connection and probably will be very useful for this research. Okay, we will talk about it later. And okay, today I'm bringing you an island story of these beautiful and cute creature, green-spotted grass lizard, in a small island around Taiwan. Okay, so first, let's check what is island rule. It's a classic evolutionary phenomena that proposed last century. It states that a big animal will be smaller on the island and small animal will be bigger on the island. And the main driving force is competition and predation on the island. And it's generally occur on land vertebrate, including mammal, bird, and especially reptiles. So the size would shift, would change on the island, and not just the size. The other rest of the phenotypic traits would also change. It is called island syndrome. It states that the small animal on the island will be not just bigger, they will reproduce larger offspring, has longer lifespan and reduce aggressiveness, their coloration, and therefore reduce sexual dimorphism. Okay, this is island syndrome, and it gets more and more attention in these, I think, 5 years. And we believe it happened on our target species, green-spotted grass lizard. It's a small species with this size and very, very long tail. Okay, it's distributed in North Taiwan, including the small island I studied. And the male will show intense green spots on their body side, only male in Taiwan. Okay, and then usually they have very weak or almost no territorial behavior. And we know these species so well because previously we did a long-term capture model capture study on these species in Taiwan. This is what we usually do at one single night. Usually we can capture more than 200 individuals at one night. They are daily active, but they were sleeping on the grass, very easy to capture at night. So that's why we do that. Okay, so we do it monthly for 10 years. So we have a lot of data that we know their population and their life history. And believe me, when you have such a number of sample size, it's very, very good when you capture it. But when you have to handle it or analyze it, it will be quite hard. Okay, so in short, they are very, very strategical species. They reproduce in the early summer, and then the hatchling will grow up very fast, then join the adult generation, and then reproduce next year, and then die out. So they are very, very, they have very short lifespan, only around 1 year. Okay, but it is the situation in Taiwan. Probably not in this island. It is Guishan Island, very small and close to Taiwan. And it's only 7,000 years old. So in evolutionary scale, it's extremely short. Okay, 12 years ago, I visited this island, and as most herpetologists would do, we herping. So we just capture everything on the island. And when I captured that, I immediately felt it's a bit different because it was like holding a different species in your hand because it's too big. So I keep these interesting phenomena in my mind. And 2 or 3 years ago, I take it outside because I became a PI, and then I wrote it as my first proposal and get the funding. So it's a lucky system for me. Okay, so in this study, we aim to know if they have island syndrome in several aspects. So we sample them on this island and the other two locations as comparison. And we quantify their morphology, reproduction, and their behavior to know their aggressiveness through skin injury and toe missing, and then quantify their side color, like this. We use SHV system and a standard photograph to quantify their color, to know their hue, saturation, value, and the area of the grain. Okay, this is how we measure their ornament. Okay, so I'm not going to too detail about the method. Let's just check the results. And here, this is the result of the body size. X-axis is the population, Y is the body size, length, and mass. So you can see that in Taiwan, the maximum body size is around 55 millimeters. But in that island, a lot of individuals bigger than the maximum record in Taiwan. It's happened in the length and the body weight. Averagely, it could be 20% bigger than the population in Taiwan. So they are indeed bigger. And not just the adults, the juvenile, the newborn is bigger too. In here, the same population and size. In that island, it could reach around 50% heavier than those born in Taiwan. The hatchling on the island just look like perhaps 2 months or 3 months old juvenile in Taiwan. It's very, very different. It's huge, and the egg is huge. So they reproduce larger offspring. And let's check the population structure. This is the situation in Taiwan. In the early breeding season, as you know, there are strategy species. Most of the individual will be ready to reproduce. But on the island, it's very different. The generation overlap were higher, and according to the body size, it's easy to imagine they have longer lifespan. And in the lab, we keep them in the lab. Several individuals still alive over 3 years. So we think they have longer lifespan, and they're more case strategical. That's some good part which fits island syndrome's prediction. But the rest will be more exciting because it's totally outside. This is the behavior results. The proportion of injury and the proportion of missing toe. And you can see that in Guishan Island, they are more easy. They are easier to get injury. And in the lab situation, they usually bite each other frequently. Compared to, it's usually not happen for the Taiwanese people, Taiwanese population. So they are more aggressive. And they also more colorful. And here, the light circle is Taiwanese population, male and female. And the dark circle is male and female on the island. Let's see the Taiwanese situation. These two circles, the light circle, separate to each other. So they are very different in their color. Clear sexual dichromatic in Taiwan. But on the island, it shifts to the left. It means greener, more saturated, and the area of the grain larger. And most importantly, there are a lot of overlap between male and female. Some female looks just like male. The sexual dichromatism decreases on the island. Let me show you some real picture that you can feel it by yourself. This is the Taiwanese male and female. This is what happened on the island. Yeah, it's totally different. It's more colorful, it's bigger, and even females show the green spots. So this is what we know about them. They are bigger, they reproduce larger offspring, and they are more case strategical, which fits island syndrome. But on the other hand, they increase their aggressiveness and coloration, which upside to island syndrome's prediction. So this is what we know about their phenotypic difference. We know what happened in this system. And the next will be try to answer how and why. So currently, we already finished the quantification of the environment, and we will probably do this genetic analysis next year to know the short-term and long-term concession of this system. However, all of it is still in single species situation. I want to extend it to other, but it's it's impossible to sample, to capture every species on the island and on the continent in short time, right? So it's impossible, but the collections will be very useful right now. So in National Museum of Natural Science in Taiwan, we have at least 15 species of this genus. Currently, there are 55 species in the world. We have 15, so probably it will be not enough for me to check this interspecies situation. But however, I need something even inside the single species. I want to compare the population on island and on continent. So luckily for me, I found some museum has these collection. It's collected by Van Denver and Thompson and in whole East Asia area. And now it's in CAS. So that's why I emailed Raina and Lauren last year and participate the Spnhc last year. We know each other, and I would lovely to go to CAS again to measure these valuable specimen for me. Okay, and in the end, of course, if any one of you know grass lizard specimen on continent and on the island, please contact me. Okay. And final, I need to thank a lot of institutions that helped me to assess the sample size, and of course the money is the most important thing for research. And the group of herpetologists in our museum is very good. Without their help, I cannot do this research. And final, thanks for your attention.
Thank you for bringing us such a wonderful and lovely stories. And we have 3 minutes. Any questions for Zhang Wei? No, as usual. Okay, please.
Thank you for a very interesting talk. And then, could you, if you know its geological history of the small island, I forgot the name of island, has been connected to the Taiwan island before?
No, never. It's
Never.
Volcano Island. Yes. But whatever is very close to Taiwan, only 10 kilometers. So very, very close. Even human can swim today. Yeah, maybe if you are strong enough.
And sharks.
Have you measured the predation pressure in the two different?
Yes. I think I have. Yeah. Oh, yeah. I measured the predation pressure through the clay model. Okay. So we put it, I think 100 of them for each sample size in each season. And we found that it's quite similar. Currently, we don't finish the analysis yet, but according to the raw data, it's very, very close. So currently we still don't know what happened in the ecological environment. Yep.
Are the predators the same species that, based on the marks like birds or?
You mean the predation pressure is?
No,
similar?
the predators.
The predator should be very similar because the major predator is bird, especially the egret. So they fly to everywhere, yeah.
I know you said you're still collecting data, but do you have any hypotheses for why there's enhanced aggressiveness and enhanced dichromaticism on the island specimens?
Okay. Yeah, I do have some hypotheses, but probably so far it's hard to examine it. Currently, we know the environment very similar, and genetics, I think it will have some difference, but I believe it's caused by something called ocean filter, that only strong and big enough individual could cross the ocean. And then once they arrive at a new island, it's kind of bottleneck, but has some direction. So it will be totally different. That's why they could be so different in this island within the very, very short time. That's what I think. And we also do some common garden experiments, and we try to reproduce the F2. Currently, we have F1, and F1 are different. So I think it's genetic based. Yeah, not statistics. Yeah.
Okay, it's time. We thank all the speakers in this section, and it's time. And also thanks for everybody's attendance. And the next section will be 4 o'clock, and it's time for the tea break. Thank you for your attention.